keep your eye on the ion exchange chromatography. So I've talked before about chromatography, and it's this way that we can purify proteins out of like a solution containing a bunch of different proteins. So we take that solution with all those different proteins and we flow them through these columns, and these columns are filled with these little bitty beads called resin. We can use different types of resin with different types of properties to separate them based on those properties. And so with ion exchange chromatography, we're separating them based on their charge. So basically those resin, they're like those little like beads that fill in the column. And they're either positively charged or negatively charged. And so you choose depending on your protein charge, which we'll talk about more in a second. But then you bind it to the column, and then you like wash it off with like increasing amounts of salt, or you change the pH, and that's gonna, so if you change the pH, you change the protein's charge, so that can make it like not stick, because you want it to stick based on like opposite chargedness. So if you have a positively charged protein, you're gonna want to use negatively charged resin. And we call that cation exchange, because we're exchanging the cation. So the cation is the positively charged thing. We're going to talk more about all of this, don't worry. But so if you have, so that would be cation exchange, and then anion exchange is the opposite. So if you have a negatively charged protein, you have these positively charged beads. And then what happens is you get your protein stuck to that, and then you either change the pH to change the protein's charge so you don't have the opposite charge stickiness, but that could also like make the protein unhappy. So typically what we do is we use a salt gradient, or like a stepwise salt elution. So elution is when it comes off the column, but basically when you increase the salt, it's going to kind of like compete off the protein, because salt is actually made up of like a positive part and a negative part. So salt's just like a neutral combo of like a cation, so a positively charged thing, and an anion, a negatively charged thing. And so when they come together, it's neutral and you get this salt. So, but when you dissolve the salt, then they come apart, and then you get this positively charged thing and the negatively charged thing. And then those things can compete with your protein to get off the column. So before we've talked about like affinity chromatography, we have like, usually when we're talking about affinity, we're often like talking about like having a special tag, like a strep tag or a his tag, and then you have resin that matches it. So you'd have like a streptactin resin, or you'd have like a Ni-NTA resin that would bind the his tag. So we talked about those things in the past. And so this is similar, except that we're using something natural about the protein. So we don't have to like add an extra tag on it, because we're just separating it based on the protein's charge. So it's not, typically we do like that more specific affinity chromatography first, because only your protein's going to have that special tag, because you put the tag on there. But so it's more like selective. It's just going to like pull out your protein, and then like anything that's just like kind of happens to bind, or that's kind of stuck to your protein or whatever, that sort of thing. So with this method is more like generic, like you can use it for any protein. It doesn't even have to be like a recombinant protein. You can use it if you just try to like purify a protein out of a cell, tagless and all, whatever. But it's less, there's gonna be more proteins with a similar charge. So you're gonna have, so this is typically used like after you have that typical like capture or affinity chromatography step. Then you go to this to kind of like purify it further. And then you often then go to a size exclusion, which is gonna separate things by size to get rid of things that had a similar charge but different sizes. So you basically take advantage of different properties of the protein to try to separate them based on the mixtures. And so in each of these types of chromatography, you just have like different types of resin in your column. And so this is like my favorite column, and I'll show you more why I love it later. But it's a mono S column, which is actually like discontinued, and now they have this like different like capto thing resin, which I'm kind of terrified to try, because I'm like so close to graduating, and I like don't want anything to change and mess anything up. I want to keep everything the same experiment wise. So I'm working that column to its limits, and it's really helpful for me because I'm trying to separate this protein that binds its positively charged protein. So I use this mono S column, which is it's negatively charged. It's this cation exchange column. So I'm exchanging the salt that was like on the column for the protein, and then pushing off the protein with the salt. And so it's like this cation exchange, because I'm exchanging with like the positive part of the salt. So like in my case, it's like potassium. That's the positive part that's gonna push off my protein, and then my protein, yeah. But anyway, so the protein I study binds to nucleic acids, and nucleic acids are like negatively charged. So that's going to lower the charge of the protein. So it's going to be less negatively charged if it's bound to the protein, to RNA, and so that's going to let me separate the RNA bound from the non-bound, because the non-bound is going to like take more salt to compete it off. It's really cool, and I'll show you later. And I should like get back to the basics and telling you more about these things, because the way you choose like which column to use, like do you want to use the positive charge, do you want to use the negative charge? Well, it depends on your protein, right? So like if I were to use the anion exchange column, I'd be like screwed, like it wouldn't work, like because my protein just wouldn't stick. And so we want the protein to stick, and so we have to figure out like how charged the protein is. And so this is something that has to do with something called the PI, or the isoelectric point, which is the point at which a protein is like neutral overall. So we'll talk way more about this. But pH is like a measure of protons, and we can like so amino acids are protein letters, and proteins have different chains of these amino acids, and some of them can be positively charged and some of them can be negatively charged depending on the pH. And so because proteins have different numbers and combos of these, they're gonna have proteins are gonna have different charges, and so we can actually use that software I was telling you about yesterday, Program. It's like this free online tool that you can use to calculate the theoretical PI, but it's gonna be like, it's theoretical, it's not like exact. But anyway, we'll talk way more about this. And sorry, I'm just like trying to rush through things just to try to give you an overview of what we're gonna talk about. So I'm gonna talk about things in like a more formal way, but I'm also going to talk about in a more fun way, because really, like, I like thinking about ion exchange chromatography and isoelectric points as proteins being chains of Cookie Monsters. And so I know some people don't like when scientists anthropomorphize, but what about Cookie Monster and Cookie Monster porphize? I don't know. Okay, let me explain what I'm talking about. We'll get back to this like more serious looking view. But how I actually really like to think about things is kind of like a Cookie Monster. And so let's step back and think about things like a Cookie Monster. Okay. So you can think of pH as a kind of measure of proton jar fullness. So instead of protons, we're not going to think about cookies, okay? So a proton is a cookie, and it's an H+. pH is a measure of how full the cookie jar is, how many, like the concentration of protons. But so like that bracket means concentration of, and so if you look at the pH equation, it's actually like a log 10 of the of 1 over the number of protons. So a lower pH is actually going to correspond to more protons. So the more protons you have, the lower the pH and the more acidic. The less protons you have, the higher the pH and the more basic or alkaline. And so if you think about it, the pH is going to depend on how many protons you start with, and how many like Cookie Monsters, so like acids or bases there are, and how greedy they are. So if you imagine you have like so the pH is at the solution level. And so the pH of a solution is going to depend on the concentrations and strengths of the acids, the proton donors, and the bases, the proton takers it contains. So imagine you have this cookie jar that's partly full and you invite over some friends. So some of the friends are going to bring cookies to add to the jar. So they come with cookies. So those would be like acids. So they have this proton that they can donate. So if you have a strong acid, it's going to give that cookie away no matter how like full the jar is. But you also have like weak acid, and so they come with the cookie, but they really want that cookie, and so they're not really going to give it up unless the cookie jar is really low. And so they're kind of like, I like to think of it as they're kind of like, they're not as generous, so they're like, well, there's still some cookies, so if people wanted a cookie, they could take a cookie. But if the proton jar is really low, then there's not enough cookies for everyone to take one, and so they'll be nice and give it up. That's really not what's happening. What's happening more is that it's like a concentration thing, and so if it gives up a proton, it's less likely to find another one to bind so it has like a weaker hold on that proton, and like if it lets it go, it's harder to find another one to bind to. Whereas if you have like a high pH when there's when you have a low pH when there's tons and tons of protons around, then if it like drops its cookie, it can get another one. But I like to think about things more just like thinking like a Cookie Monster. So that's what we're gonna do. Okay, so the bases come cookie-less and greedy. But how greedy? So here's the kind of weird thing where things get kind of complicated with acids and bases, because if an acid gives up its proton, now it's kind of a base. So we call it a conjugate base. So it's like a conjugate acid, conjugate base. So once the acid gives up the proton, now it's a conjugate base. And so, but you can, so if you think of, so the bases, but some bases are like more, like some things are more like acidic, like once they give up that cookie, they're not going to want to take it back. Like, so if you have a strong acid, like it's not going to want to take it, it's not going to be a good base. If you have a weak acid, it might be an okay base because it's kind of iffy about whether or not to give up that cookie, right? But some things are really like basic, and so they will take a cookie even if the jar is really low. Like they don't care, like who cares about everyone else, we'll just take a cookie. Remember, that's not how things work. It's really that just that they have like, they're gonna bind to a titer and that sort of thing. But I like to think of it more like they're really greedy. And if they're a weak base, they're like, okay, well, other people might want them, so I won't take some until like everyone's taking some and there's a lot left over and that sort of thing. And so remember, pH is how full the cookie jar is. And so at the end of the night, how many cookies are in the cookie jar are going to depend on like how many of each type of friend there are. And so when we think about a solution as having like acids and bases in it, well, when we think about acids and bases, like you might be thinking of things like NaOH, so like lye and that sort of thing. But proteins are actually made up of like chains of where some of the residues, so some of the amino acids, like when amino acids are protein letters, when they combine, the leftovers are the residues. And so some of these protein residues can act as acids or bases, and that allows them, so when you have this like linked up protein, most of the residues are going to be neutral, but some of them are going to be like cookie monsters. And so some of those cookie monsters are going to be basic and some are going to be acidic. And so depending on the pH, so depending on how full the cookie jar is, and how greedy your cookie monsters are, and how many of each of the cookie monsters are in your protein, then the protein's going to have a different charge. And so you can hopefully see now that proteins are going to, because they have different combinations of cookie monsters and numbers of them and that sort of thing, they're going to have different charges, and that charge is going to depend on the pH. So then overall, we can define a point at which the protein is neutral overall. And so this is the isoelectric point, or the pi. And so the pi is the point at which a protein is neutral overall. But there can still be like some positive and some negative charges. So let's step back. And so we've talked about pH and we've talked about pi, but the pi is not just going to depend on the pH, it's also going to depend on so the pi is going to depend on the composition, so like the numbers of these different sometimes positive ones and sometimes negative ones. So let's talk more about those ones. So we have the strength of not all like acids and bases are alike. And so we've talked about how like can have greedier cookie monsters or like more generous cookie monsters and that sort of thing. So there are really 2 sometimes negatively charged amino acids and 3 sometimes positively charged ones. But really, the 2 of them are almost always negatively charged, and 2 of them are almost always positively charged. And histidine is the really one that's kind of like in the middle. And so, how do I know this? Well, what I'm showing you here, so this is like the pKa. So here you can see the different forms. So like aspartic acid, and then so it has this carboxylic acid group. When it acts like an acid, gives up that proton, and now it's aspartate. So it gave off that proton, and now it has this negatively charged group here. So it's a carboxylate group instead of a carboxylic acid group. So this is why you might see sometimes it written as aspartic acid, sometimes it written as aspartate, sometimes written as ASP or D, which are its abbreviations. And then for glutamic acid, you have glutamate, same type of thing. You have this deprotonation, gives you this carboxylate group, and its abbreviations are GLUE and E. So, and then for lysine, you have this, your protonation is happening. So it's taking a cookie, and that's going to make it positively charged. And so this is, sorry, this one's drawn backwards. The positively charged should be over here, and the neutral should be over here. But that's wrong. Sorry. So the protons, and then histidine, it's kind of in the middle. So what I'm showing here is like the point at which you go between one or the other. Well, for each, if you're looking at the individual level, so if you were looking at an individual amino acid, you couldn't say like exactly at a pKa of 6 it would switch. But basically the pKa is like when it's half likely to be protonated. So like if you had a ton of histidines at a pH of 6, half of the histidines would likely be protonated and positively charged, and half would be deprotonated and neutral. And so you have this pKa is this measure at the individual acid level, and then Pi is the halfway point, or like the neutral point for the entire protein, and then pH is a measure for the whole solution. So the pH is the proteins will contribute to the pH, but so will everything else that's in the solution. And so going back to our Cookie Monster and the idea of the pKa, so below the pKa, so the pKa is like a measure of the Cookie Monster greediness. And so above the pKa, there's not that many, that's where like the cookie jar is running low, and so the protein or the acid's going to be like, okay, you can have mine. I see there's not that many. And then below the pKa, the protein's like, or the acid's like, the jar's full, I'm gonna keep this one. But they have different points, like tipping points, about like how low the jar has to get in order for them to give theirs away. So if you have a strong acid, it's going to give its away even if the jar is pretty full. And so the pH at which it's going to give it away is going to be higher. And so it's going to have a higher, it's going to have a lower pKa, and it's almost always going to be above its pKa because the protons, the more full the jar is, the lower the pH, remember? So it's going to have a stronger acid is going to have a lower pKa, which means that there's more protons in the jar and it's still going to give its up. But if you have a weak acid, it's going to have a higher pKa. So you can be at the cookie jar will have to be less full before it'll give it up. So it's a little greedier. It's going to want to hold on to that cookie more. So yeah. So if you look below the pKa, molecules are more likely to be protonated because there are lots of protons around. At the pKa, it's 50-50. So it's like on the fence about whether to donate. And above the pKa, it's more likely to be deprotonated because there are fewer around. And different acids have different pKa's. So the greedier, the weaker at the acid, it waits until the jar is running lower because remember, the acid, when something acts as an acid, it's donating a proton. And the lower the pKa, the more generous, so the stronger the acid. It donates even when the jar isn't that low. So this is happening at the individual acid level. And so if we go back to our protein, it's happening in these individual Cookie Monsters. So the pKa is describing it at this point. So at the level of the individual amino acids. But remember, those are just individual Cookie Monsters in this long chain of proteins, long chain of amino acids in your protein. And so the protein charge overall is going to depend on this whole contribution from all of the different Cookie Monsters, and that's why you get the Pi, which is the measure at which the protein is neutral overall. So if we go back to the protein, if we want the protein to be charged, So if we use a cation exchange resin, we're expecting our protein to be positively charged. And so we want to use it when there's plenty of protons around. So when there's a lower pH. So we're going to use it below the pH, the protein's pi, because the pi, remember, is the point at which the protein is neutral. So it has like an equal number of positive and negatively charged parts. So below the pi, the cookie jar is fuller than it's than like the threshold or whatever, and so it's going to take a cookie, and so you're going to have a positively charged. With anion exchange, you want your protein to be negatively charged, and so you want to use it above the protein's pi, because remember, the higher the pH, the lower the number of protons, the lower your cookie jar. So there's less cookies in the cookie jar, so there's fewer cookies that it can take, and so it's going to be negatively charged. And so, but remember, different proteins are going to have different pi, so different points at which they're going to be neutrally charged. So different, like, because they have different combinations of those amino acids that will give or take and are more or less greedy. And then, so the basic idea is we get the protein to stick, and then we can peat it off with salt. And so ions are just charged particles, and so they get their charge because they have uneven numbers of protons and electrons, which are these little subatomic particles, so like smaller parts of atoms. And so cation is just a term we use for a positively charged ion, and anion is a negatively charged anion. And salt is just a neutral combo of ions. So if you have like NaCl, like sodium chloride, which is table salt, it has this positively charged sodium and this negatively charged chlorine, and then when you dissolve them, they like come apart, and so then the positively charged parts can act as like act as cations, exchanging for the protein, and then like the chlorine in the case of anion exchange exchanging. So you can use like salt for either way, because it has both parts. But we're going to stick to the column based on the idea of opposite charges attracting. And so the more oppositely charged a protein is, the stickier it's going to be for the resin. And so the more salt it's going to take to exchange off the column. And so if you were to do like a salt concentration gradient, you would see that the least oppositely charged protein comes off the column first, and the most oppositely charged protein comes off last. And so, but you want to, so you're going to have to so you can do this to try to like separate proteins based on their different charges if you have a solution containing different proteins. But you want to make sure that you're using the right kind of resin, so you need to know like what charge your protein is likely to have. So you want to figure out the protein's pi. So yesterday I showed you how we can use this tool called XPazy-Prot-Param. So you can also access this from Uniprot, but I want to do AGO2 human, which is this protein that I study. Okay, so if I go here, so I want this is the full chain. You can also look at like just parts of the chain, and it's going to calculate a bunch of different things. So number of amino acids, molecular weight, and then this is what we care about now, this theoretical pi. So it's 9.32. So this means that its neutral point is going to be 9.32. Any pH below that is going to be positively charged, and any pH above that is going to be negatively charged. So typically, I do my experiments at like a pH of 8, which is below the pi, so my protein is going to be positively charged. And therefore, I'm going to want to use a cation exchange resin because my protein is going to be a cation. It's going to be positively charged. And so just a note, we often talk about when we call things like positively charged, or in biochemistry, like when we're talking about proteins and stuff, so we would often call this like it's basic. And that's because it's acted like a base and it all like taken up a bunch of protons, and so now it's like the conjugate acid form, but because it acted like a base at like the normal conditions or whatever, now we call it basic. And so that's why when we were talking about like these residues. So we call these like these ones like acidic amino acids, because they act like acids and they give up the proton. And so in the form that they're normally in, they have acted as an acid already, and so they're negatively charged. The basic ones, they have acted as a base and taken up a proton, so they're positively charged, and now they're the conjugate form. But so don't get confused by that. And so you can see why I said histidine was kind of iffy, because its pKa is about 6 and body pH is about like 7.4 or whatever. And so it's kind of iffy because it's on this borderline, because remember the pKa is the like half likely to be point. So it's halfway likely to be in like either form. So if you have like a whole bunch of histidines, like half of them would be plus and half of them would be minus. Or if you were to look at an individual, you were to go pick a histidine, it would have a 50-50 chance of being positively charged. You also see here I'm drawing this like dashed line, and this like, because this charge is actually like delocalized among these different groups. But anyway, so when you look in like the body or whatever, you're more likely to find the aspartate glutamate forms and like the positively charged lysine and arginine forms, because the acid ones are below their pKa or above their pKa, and the basic ones are below their pKa. But histidine is near its pKa. But just because they're normally that way doesn't mean they're always that way. And so pH is a measure, like it's more like a global level. But in the context of a protein, these amino acids, like the pKa, can be shifted. So these pKa's like are technically measured like with the free amino acids, so like not connected to a protein. But in a protein, you can kind of have like pockets where the pH is slightly different, like a local pH, and so that can influence whether it's positive or negative. Or you can also even have like things nearby, like trying to pull it away and that sort of thing. So aspartic acid and glutamic acid and whatever, the lysine, like the non and the forms or whatever. So you can actually sometimes find the different forms, at least temporarily. But histidine really can go back and forth more readily. But going back to the idea of chromatography. So we've talked about this like acid base, and we've talked about like how to find the PI, and so now we want to actually use it. So chromatography we've talked a lot about. It's like you have these columns filled with these resins with different properties, and then you flow a solution containing protein through them. And then it separates the proteins based on how they interact with the resin. And so we talked about affinity chromatography, and now we're talking about ion exchange chromatography, and I've also talked about size exclusion chromatography, I think, in a video, but I know I have posts on it. So that's going to separate things by size, and that one you're not actually sticking, you're just going through like different tunnels in the beads, and so the smaller beads have to go through more tunnels, and so, or smaller proteins have to go through more tunnels, and so they take longer and they'll elute later. But I want to show you an example of like ion exchange chromatography, so like how I use it. And so I set it up on the Acta, which is that machine that I was standing by in the beginning, and I've done a videos on that. But I have these two buffers. So one of them is no salt and one of them is high salt. And so the buffer is just like a pH stabilized salt water. And remember, the pH really matters here. So you want to make sure that the pH, at the pH you're using, your protein is going to be at the charge you expect it to be. And also another note, near the PI, like, so when a protein is neutral, it's likely to be least soluble, although it's not always true. And so the reasons why you don't want to be like at the PI or whatever. And you also don't want to be like too far in either extreme, because that can make the protein unhappy. But yeah, so I have this like no salt and high salt, and then I set it up so that it mixes them and it does this gradient. What I actually do is like I do a stepwise gradient, because I know what concentration I need to get to separate the proteins that I want. But you can normally, if I'm like just starting out or something, I would do a concentration gradient. It's really like a special situation with my protein. And then they're gonna come off, and you see this is like the chromatogram. So this is showing like the UV absorbance as the protein goes through that UV detector, and then it's going to like dispense it into this fraction collector. And so I do this into like a 96 well block,
And so then each of these wells is going to have like, on my chart, it'll be like, it'll have these like tick marks that are telling me like the different fractions. And then I go, okay, so I want this protein, and I'll come down and I'll take those, the block, go look through the blocks and find those fractions, and then take those and then move them onto the side of exclusion. So the reason why like this is my favorite, oh, and just another note, so sometimes if you're having problems with ion exchange chromatography, you might want to change like the pH or the salt concentrations. So like if you have like poor binding or whatever, you might need to change the salt or the pH in different ways depending on what type of exchange you're using. Sometimes you're not really sure, like the protein is kind of like being weird. This happened to me once. I had a protein in like, because remember it's a theoretical PI, so it's not always like totally accurate. It's going to depend on things and that sort of thing. So I actually like hooked up an S column and a Q column. So the S column is like a cation exchange column, and the Q column is an anion exchange. So like I ran them, like I connected them and ran it through both. Then took them apart and then looked to see what bound to each of them to try to figure out where it was. If your protein's really uncharged, you might want to like try like a hydrophobic exchange column, which might help. But I'm not gonna go into that here. But I want to show you like how my purification process. Okay, so I've told you all about AGO. So if you've been following me, you know what this protein I'm talking about. If you haven't, then go back and learn about it, but I'm not going to bore everyone again. But AGO is like short for argonaut, and it's this protein I study that's involved with RNA interference, so it uses these like small RNAs, like microRNAs or siRNAs, to target and messenger RNAs for like degradation or repression and stuff, and it's really cool. But for now, you just need to know that it's a protein that binds to short pieces of RNA. And so I express it recombinantly. So I take the gene for human argonaut 2 and I recombine it with this like gene in like this back mid, so it's like this insect virus, and then I infect insect cells and get those cells to make the protein for me, and then I purify it out. And so within the protein, when the insect cells make it, so the insect cells have their own like they have their own like small RNAs in the insect cells. So most of the AGO is going to actually just like bind to some of the insect cell small RNA. So it'll be like endogenously loaded, and then a little bit of it will be RNA free, so it's not bound. And so this is the form that I want for all my experiments, because I want to be able to test its binding and that sort of thing. And so to make this purification easier, so for both the endogenously loaded and the RNA free, like I can't tell it to make more of the RNA free or whatever, and it's all the same. So it all has this like sumo strep tag. So the sumo is just like a fusion partner that's going to help with solubility and that sort of thing, and then the strep is like an affinity tag. So I'm going to use a strep tag purification. So I have this column with this resin that'll bind to the strep tag, and then I can wash everything off and then compete it off with this thiobiotin, and I've done more about that, so I won't talk more about that. But now I'm done with that tag, so I'll cleave the tag off. And so now my protein doesn't have the tag to separate by, so now we have to switch to separating it by things that are natural about the protein. And so next step I do is this one I talked about today, which is this cation exchange chromatography. Okay, so here what I'm showing you is on the left, so that green line, that's the salt concentration gradient. So I'm using this stepwise elution, whereas I have this wash. So when you want to, so first things first, like when you have a column, you want to equilibrate it at the pH, like in the salt buffer that you have, and so that the salt is going to like stick on there. So the column is going to be covered with the salt, and then it's going to be covered with your protein, and then it's going to be covered with the salt again. So you want to make sure that the column is like all covered with the salt of what you want. And so you want your protein to stick, but you want to make it so like you don't want you don't want too much other stuff to stick. So you have to play around with the salt and that sort of thing. And then you want to wash through like all that stuff. So this kind of over here you have like a wash where you're trying to wash off anything that's not stuck or that's just weakly stuck, because your protein should be strongly stuck. And then you up the concentration. And so before I showed you like a linear gradient, and here it's the stepwise elution. So elution, remember, is just coming off, and this is the chromatograph, and so it's showing you as it goes through the detector into the fraction collector, the UV. So like I have it two things. So I have the UV 280 and the UV 260. So normally when proteins absorb most strongly at 280, and then nucleic acids, so like DNA and RNA, are 260. And so you can see here that the 260 is above the 280 in this one, so it's the part that's bound to the nucleic acid. And this part here, it's RNA free, and so you have the 280 over the 260. More on this in a minute. But now I separate this endogenously loaded from the RNA free, and then I take that through gel filtration to get it. So gel filtration is just another term for size exclusion chromatography, and so now you can see I've gotten it even purer. Then I can use that for my experiments. So how do I know it's RNA free? Well, I told you before, like, so DNA and RNA, they absorb at different wavelengths. They absorb most strongly at different wavelengths. So they overlap. Their absorbance spectrum overlap. So the absorbance, I've done more on absorbance now, so I'm not going to go too much into it here, but just know that their absorbance overlaps, but the 280 is going to be predominant. It's like more specific for protein. And then the 260 is going to be nucleic acids. And so that's how I knew when the one curve was above the other and that sort of thing. And I also know because if I run like a urea page gel, so you might be familiar with SDS page, which would separate proteins. Urea page is kind of similar, except it looks kind of similar, but it's a different kind of gel that we can use to separate RNA fragments by their size. So it has like a tighter mesh, and that's what we can make this really tight mesh to separate these like short RNAs. And so on the left I have just like a size marker, so a microRNA that's 22 nucleotides long, and then I have I've loaded the same amount of like endogenously loaded fractions, so this first peak, and then this RNA free like the second peak. And you can see that the endogenously loaded already has, it really is endogenously loaded, like it has some small RNAs around the size of a microRNA. Where this the RNA free, it doesn't have any yet. So this is just stained for generically for nucleic acids. And yeah, so that was kind of like a whirlwind tour about pH, pKa, whatever. So yeah, and going back to this like prop param, like it doesn't know, like it's theoretical PI. It's obviously not, it doesn't take into account that it has sometimes has RNA or not, because it's not going to know. So yeah, you can use this tool, and the cool thing about prop param is that, so I just put in a gene name, but you can paste your own amino acid sequence here. So if you had like a different version of a protein, like you made changes to it where it still has a tag on it or something that would affect the chart, the PI, and that sort of thing, you can find it out here. And so yeah, and it's just this free online tool. So I highly recommend it. And I will direct you to like posts with more text and graphics and that sort of thing, and I'll also go fix those graphics that have the problem. So sorry about that. And yeah, so hope that helped you, and happy purifying.